Beautiful.
Good morning, everyone. Hi, hi. I think we've just got a few more people coming in to the Garvan auditorium. Great to see lots of familiar faces and a few newbies as well. We're also joined by around 100 people in the Zoom room, so welcome to everybody in the Zoom room. And of course, we will be streaming via Facebook Live as well. It's a great pleasure to host today's event to mark the International Day of Women. Before we kick off, I'm humbled to acknowledge the traditional owners and custodians of the lands on which we meet, the Gadigal people of the Eora Nation, and pay my respects to elders past, present and emerging, and extend warm wishes to any Aboriginal or Torres Strait Islanders joining us in the room in person or via Zoom. Welcome to you all. For those of you who don't know me, my name's Mara-Jean Tilley, and I've got the great privilege of heading up the Garvan Research Foundation, which is the fundraising and marketing arm of the Garvan Institute. And we've got a very, very talented lineup of female scientists to speak with you today. So really excited to have the opportunity to introduce you to just some of Garvan's incredible women scientists. In the highly unlikely event of an emergency, please do follow my instructions at all times. We would exit in the unlikely event of emergency via the front of the auditorium or the rear of the auditorium. We do have bathrooms located down here on the ground floor for anybody who needs to use the facilities throughout today's session. So I'm always humbled when we come together for these events to think of those who came before us at the Garvan Institute and here on the St Vincent's precinct. And of course, Garvan and St Vincent's Hospital were founded by an incredible cohort of women, the Sisters of Charity. The Sisters of Charity had the vision to not only continue treating disease, but to understand the underlying cause of disease so that we might better diagnose, treat, and ultimately prevent. They also did a number of other firsts here in Sydney. They established the first wards for HIV patients just across the road. They established the first heroin injecting rooms down in King's Cross. And they've got a number of other firsts behind their legacy. So we stand on the shoulders of the Sisters of Charity today. The format today is a little bit unusual, so we're going to do a moderated Q&A, and then we will invite questions from both the audience in person and via the Q&A chat function in the Zoom room. So those of you who are here, if you have any questions for our talent, please pop your hand up and we'll get a microphone to you. Those of you in the Zoom room, feel free to pop your questions in at any time. So, to our talent. I'm thrilled to introduce to you Associate Professor Sarah Kummerfeld, our Director of Data Science. Welcome, Sarah. Our Senior Research Officer for the Matrix and Metastasis Laboratory, Dr. Amelia Parker. Welcome, Amelia. We have with us Dr. Venessa Chin, a research officer in the Powell Laboratory, but also a medical oncologist here at the Kinghorn Cancer Centre, which many of you will know is a joint initiative between the Garvan Institute and St Vincent's Hospital. And last but not least, Associate Professor Elissa Deenick. Elissa is the laboratory head of the Lymphocyte Signalling and Activation Lab. That's a tongue twister for first thing on a Wednesday morning. Welcome, ladies. Sarah, I'm going to start. Sarah, I'm going to start with you. Tell us a little bit about your work and what inspired you to come to Garvan.
So as Director of Data Science, I oversee both the research side, data science, which is working out how to get the most out of our data. So biology and medical research now is a big data science. We're not able to just use Excel or lab notebooks to be able to analyse the data we generate. We generate petabytes of data. And when it's at that scale, you need people who understand computation and can program and can use machine learning and AI and all of these types of technologies that you think about in lots of other disciplines to be able to make sense of their data. And so we have on our team not only people who understand the medical side and the biological side, but also people who have maths, statistics, computer science backgrounds. And so we've got researchers who are developing new methods to analyse data. And then we also have a large team of people who do the enabling work to be able to process that data. Things like how do you actually run computers at that scale, whether it's using infrastructure that we have on site or commercial cloud. And how do you help researchers to process their data so that they can focus on research questions? So it's pretty broad. My path here, before coming to Garvan, I spent 10 years in industry at Genentech, which is the biotech company in the San Francisco Bay Area, where I worked in their computational biology department, developing new drugs and companion diagnostics.
Amazing, thank you, Sarah. Amelia, tell us about you and what attracted you to Garvan.
Yeah, I came to Garvan a few years ago now. My research is focused on lung cancer and understanding the biology of lung cancer. Part of the work I'm involved with is developing new technologies that allow us to look in a more precise and dynamic manner at the way the tumours develop, how treatments affect those tumours, and to use that information to understand which patients are going to benefit from which treatments. And one of the things that really attracted me to Garvan was that not only are they doing very groundbreaking research in understanding the tumour and the environment around the tumour, but also in pushing the boundaries of what technologies can teach us, including the work that Sarah was talking about in big data, to inform how patients will respond to treatment and help us to more effectively match the best treatment to the best patient. And the span of the research being done at Garvan, from a biology and understanding aspect all the way through to clinical trials, was really attractive to me. I came to Garvan after working at the National Cancer Institute in the US, where I saw a great breadth of research being done there across the whole scale and what a difference it made to patients as well. And I could see that the Garvan was also involved in such important work here, and that's what drew me here.
Wonderful, and that's an unplanned but perfect segue actually to Venessa. Venessa, you're a medical oncologist and you actually specialise in treating patients with lung cancer but also head and neck cancers. Tell us about what your day looks like and why you decided to become a clinician scientist, not just a clinician, which is already a very big job.
Yeah, so I split my time 50-50 between clinical practice and research. So the Kinghorn Cancer Centre, the bottom few floors are sort of dedicated to the clinics, and then the top floors are part of the Garvan Institute. So 2 days a week I see patients, I do on-call, I have ward patients, also go to St Vincent's private, and then on sort of 2.5 days a week I sit up on level 10 of the Kinghorn and try and achieve some science. And actually what we've been able to do is really try and link the problems that we face in the clinic with some of the research priorities that we have in the lab. So it's been a really, I think, productive relationship between the Kinghorn Cancer Centre and the Garvan, having people like me who can kind of span the gap, if you like, and setting up clinical research projects that involve our patients and have that loop of, you know, things that we face every day in the clinic to how can we improve things, designing research questions, and feeding those results back into the clinic. So there are a few of us now at Garvan that do that. And I think it's been mutually beneficial. The labs benefit, I benefit, and I think the patients benefit as well.
Amazing. Elissa, in addition to being a lab head, you are also running a very significant programmatic activity here at Garvan in the immunology and precision immunology space. You're the chair of Garvan faculty. You're obviously, you know, mentoring your own laboratory and scientists around the place. What does a day in the life of Elissa look like at Garvan?
Yeah, wow. I mean, different days look different. You know, sometimes it's talking to your PhD students and helping them, work through their project, which might be looking at, some patients who have an immune-mediated disease and trying to understand kind of what are the experiments we can design to really understand what's wrong with this person's immune system in the hope that down the track we'll actually be able to, treat those people better and improve their disease or maybe even cure it. So, I might be working with my students or, as part of my program, we're actually looking for clinicians and other scientists who we want to bring together to do more than I can do alone in my lab, but, to, address big questions about autoimmunity or allergy and say, what are the real questions that the doctors who are treating these patients have? Like, what do they need to know to treat their patients better? And how can we, as a program, design experiments and projects that will answer those questions so that they can treat their patients better?
Wonderful. Sarah, I'm going to ask you to go back to basics because I think we all hear a lot about big data, machine learning, AI, genomics, these really big concepts. You're talking about the scale of this exercise as being, bigger than anything that most of us can really imagine. Give us a 101. And why is it so important that we're integrating data science into the work at Garvan and medical research right around the world?
So I think probably the best area to start is with genomics. So at this point, the technologies have become cheap enough with genomics that we can actually generate a lot of data. And that's really been the turning point for biology. So if we look at 20 years ago when the first human genome was sequenced, it cost billions of dollars and it took 20 years. We now sequence hundreds of genomes a week. And so the scale of data that you're generating is just orders of magnitude different. It also means that you can just do a lot more and ask really complicated questions, things in the space that in cancer and in immunology, where you can actually ask questions about individual cells, and you can say, I want to understand what are the changes that happen in disease, say an immunological condition, at the single cell level. And then you can go do sequencing and take measurements in every individual cell and get thousands of measurements in every cell. So the scale means that you can ask questions you could never ask before. You can tackle problems that were impossible. And that's great for a lot of different areas where that complexity actually makes the difference. And when we simplified disease, we couldn't find the answer. But it also brings with it a whole lot of challenges. And so that's where the data science comes in. And we need to do really basic things like how do you move the data from this machine to the place you're going to process it? And then how do you process it? So there are a lot of pieces that you end up needing, people with different skill sets, from those who build out hardware, software development, processing pipelines, all of these different types of components, right through to the AI machine learning people. Just to give one quick example, we've got some microscopes at Garvan that can look at live cells as they're moving around. And that means you can generate videos that are huge amounts of data, but also very rich. There's a lot of information there. But to interpret it, you can't just look at it. You actually need to use a computer. And so we've got a computer vision expert now who's using similar technologies to self-driving cars to be able to understand how cells are moving around. And I think it's that key when you really get that interdisciplinary team and you've got people who bring skills from these different areas, you're going to be able to tackle problems that were impossible before.
Just phenomenal. And could you have imagined that we would be where we are now with some of this technology 10 or 20 years ago?
I was dreaming about it. So I did my PhD right at the time the Human Genome Project was completed. And so I worked on some of the initial other genomes, some other organisms. And at the time, that was pretty exciting. And we had all grand plans, but I didn't think we'd be here quite so quickly to be at the few $100 genome.
Amazing. Amelia, did you always want to be a scientist, or when did you realise that's what you wanted to do?
I very much didn't always want to be a scientist. So I have lots of colleagues where that has always been a passion of theirs. At school, I was into lots of different things. And when I was applying for university, had a very broad range of things on my list. But what really excited me initially was learning some of the details about how human biology works, and also ways in which we could use technology to help people live better lives. And that really drew me to study biomedical engineering and science at uni. And through that process, I was involved in research projects, and I just really loved the troubleshooting and problem-solving aspects of it, and followed my nose in the end to this path here. But it's certainly not something I thought I would always want to do in any way. Yeah.
Well, lucky for us. Seriously. Venessa, I mean, science is an immensely challenging career to take on. Medicine is also. What would you say has been the biggest challenge that you've had in your career to date, and how did you overcome that?
Oh, that's a big question. I mean, there's been lots of different stages, of course. I really, similar to Amelia, science was not really on my radar. And I came up to the end of my specialty training, and it became clear that translational genomics was going to be a big part of medical oncology, which is my specialty. And I felt that doing a PhD in that area was going to be beneficial to me and a good way to finish off my training. I then immediately took maternity leave, had my first child, and then arrived at the Garvan with a 5-month-old baby in the lab with my supervisor away. And I remember sitting there just thinking, so what do I do now? I've never set foot in a lab. I'd never held a pipette before. I'd never done any kind of science. I mean, my medical degree was very vocational. There was no basic science involved. And I remember another one of the PhD students came up to me and said, should we go for a coffee? And I thought, yes, let's do that. So I think that upskilling for medically trained people is a really daunting period, because we just do not have any of those basic lab skills that people who come from science backgrounds do have. So that was difficult, and I mean, Garvan has a really nice ethos in that way. Everyone knows that the medics need all this hand-holding, and actually that was a pretty nurturing environment. So that was probably one of the bigger challenges. And I think now the thing that I have to grapple with is all the multitasking that I have to do. I wear a lot of different hats, both in the medical field and in the science field, and trying to get all those things to kind of integrate seamlessly is a massive challenge. Yeah.
Amazing. And just to be clear, the baby was not involved in medical research, in case anyone's getting worried, although possibly a scientific superstar of the future, no doubt.
Hopefully.
No doubt. Elissa, immunology has changed so much. We've gone from, thinking that it's very much one thing. Now we know that it's involved in cancer. It's pretty much involved in everything. What's your big dream? How can immunology solve our health challenges of the future?
Oh, yeah, look, I mean, it really is. It's, the big new cancer therapies, immunotherapies, allergy, autoimmunity, our response to fight off COVID. This is all immunology. One of my passions, and it comes out in the title of our program, is precision immunology. And as an example, to help you understand that, think about autoimmune diseases. So I don't know how many of you have an autoimmune disease, or have friends.
Hands up.
Hands up. Anyone have autoimmune?
Disclose?
Yeah, there you go. This is a huge range of diseases. And, autoimmunity is when your immune system gets inappropriately activated, and instead of protecting you from infection, it attacks some part of your body. And traditionally, these diseases have always just been kind of treated kind of non-specifically, like, we'll just suppress the immune system, things like steroids and that kind of thing. The problem with that is that it causes a lot of side effects. It increases your risk of infection and all that kind of thing. And we also know now that what drives, this person over here's autoimmunity might be completely different to what drives this person's over here's autoimmunity. But we're using a one-size-fits-all treatment thing often. What we want to be able to do is actually identify for this person over there what's driving theirs and what's driving this person's, and then actually give them drugs that are targeted to what's driving their autoimmunity. And the hope is then that you'll be able to better turn off that autoimmune response while protecting their immune response to infection, so that that person's disease will be better controlled and they'll have less side effects and their life will be better. So that's my dream, to actually develop the tools so that we can do that, so we can give you the drug you need for your disease at the right time.
Amazing. Cannot wait for that, personally and selfishly as well. Very, very cool. Do we have any questions from anyone in the audience? Because I can talk all day, as many of you know. So I'll just do a little scan. Yes, bear with us and we will get you a microphone. So just ladies in the middle. Thanks, Dr. Brown.
Thanks to the panel, and I guess my question is directed particularly at you, Venessa, but other panel members too. If I cast my mind back a million years ago to when I was an academic, one of the things that frustrated me always was this gap between theory and practice, where research went on, but look, we don't dabble in practice, we do research, and practical people didn't have access to research or the skills to be involved with that. So it was very exciting for me to hear what you're talking about in particular, Venessa. And I wonder, for all of you, if this is a growing area where we now understand that there isn't this thing called research out there. If we don't actually know how to apply it, we're not going to get as far as you've clearly got today. Just wondered about your thoughts on that.
Excellent question. Do you want to kick us off, Venessa?
I mean, I think that idea of quickly applying medical research into the clinic is really becoming very popular. And I think that's partly because we all understand the importance of doing things that are relevant rather than just because they're interesting. But also the funding bodies are increasingly requiring that of us to have a clear plan of how our research will implement directly into the care of patients. And I guess there are advantages and disadvantages of doing that. One is that most of us have got very applied research projects with clear translational prospects, but as Mara Jean was saying, we're all standing on the shoulders of research that's been done before, so some of that more theoretical basic science research still needs to be done in order for us to make inroads into the clinic. So I think it's super important, but there's also needs to be a balance.
Maybe, Sarah, you've all kind of touched on, the evolution of genomics and the importance that is having, both in research and in the clinic. How much of this, this change that we're seeing, this coming together of the clinic and our science is driven by that genomic data that we can now get directly from patients? We can be conducting research in patients in real time with their permission. What are your thoughts?
Yeah, so absolutely. It's changed the way that we do science, because we can actually look at the population in front of us with a particular disease rather than using mouse models, although mouse models are useful too. But the other thing that it's done, and I think is a good example of application, because we're dealing with patient genomes, there's always a chance that we find something that could be useful to that patient. And so there are, if it's on point for the study we're doing, then there's a mechanism to give that information back. But sometimes you find something completely different. So one of the projects that we have underway at Garvan, led by Marianne Young, is a pathway for returning incidental findings. So it's not exactly answering your question, and it's not necessarily applied research in that specific goal, but I think it's acknowledging that we are working with patients and we need to do right by those patients. And so,
can you explain what an incidental finding might be?
Sure. So when you have someone's entire genome, you might be interested in the immune genes for the purpose of Elissa's study, but you might find a BRCA mutation, which means they're at increased risk of breast cancer. A lot of people actually would like that information back. And so when we do consent for people to do genomic studies. We give them a choice. Would you like to know about other things we find? And then if they say yes, then that information is given to them and they are put into a pathway to get the right genetic counselling and support to then have the appropriate treatment. And so it's focused on actionable findings. So things where there's something that that patient can actually do to benefit for that condition. So whether it's more screening, the case of a BRCA mutation, or sometimes a treatment right away. But I think it's a great example of where researchers are acknowledging we're doing research with some particular goal in mind, but we actually have the opportunity to help patients along the way.
Mm. Yeah, which is quite remarkable, and it means that engagement with the community is absolutely critical, and then the interface of our clinician scientists is key. So I think for Garvan, we absolutely see it as of strategic importance that we continue to attract clinicians to come and do their PhD with us. And if we're lucky enough, we get to convince them to stay on, like Venessa, and juggle these two very important roles. Rory, did we have any questions on your side of the room? Ah, fabulous. Well, we do now. We've got a microphone coming your way. And don't be shy for those of you in the Zoom room. Please do pop any questions through in the Q&A function.
I'm not quite sure whether I have the right people to ask this question, but you've got a lot of brains there. So I have a close family member who has endometriosis, has had for many years. Is there any research being done on this, or is it immunology, genomics? What does it come under?
It's a great question. I will turn to the team for their thoughts. Unfortunately, Garvan is not currently undertaking any research in endometriosis, and I myself know many individuals who live with what is both a very challenging and kind of debilitating condition that impacts hugely on quality of life. I guess, Elissa, we'll put you on the spot. What is known about it from a kind of immunological inflammation point of view?
Yeah, look, there have been, again, it's my area of expertise, but there have been some studies suggesting that there is a role of the immune system and that there's, it's driving inflammation, and that's part of the problem. But we don't know yet kind of what causes that, and how much of that is genetic, how much of it kind of is environmental, things you might be exposed to, how much of it is driven by hormones and that kind of thing. But yeah, I agree. It's a clear example of kind of a women's health problem that desperately needs more research.
And one that arguably has not had a lot of funding. So I think that there have been a few government announcements recently. I can say that we've got some wonderful clinicians at St Vincent's in this space. And I know that because I've had to, suggest informally that some of our Garvan network might go and see somebody across the road. But you're absolutely right, it really needs to be prioritised, because it's quite unacceptable that so many women live with this condition.
Can I just add?
Please.
I believe there's a group at UNSW, Associate Professor Ford, I think, and she does a lot of ovarian cancer research, but I think there's also a small bit of her research dedicated to endometriosis. But if you connect with Mara Jean, I can confirm that for you.
That'd be wonderful. Thanks, Venessa. Any other questions? Excellent.
Hello. Yeah, I don't know, like any member can answer this question. My question is that, as per I know, the lung cancer is the most common cancer in the world. And so I don't know, it might be the reason the lung cell is directly exposed to the environment. But if we consider that thing, then our eyes is also exposed directly to the environment. And so what is why this is the most common all over the world compared to the breast cancer or other like kidney cancer or prostate cancer? Is there any specific difference between?
Venessa, do you want to start, and then we'll ask Amelia to contribute as well?
I guess in all parts of our body that are exposed to the environment, we have inherent, I guess, antimicrobial processes in place to protect us. And so the eye's got excellent protective mechanisms. We've got eyelids, we've got tear ducts, and also we have the corneal barrier, which is a sort of almost impervious barrier that protects us from the environment. In contrast to the lungs, where we breathe deeply and we cannot avoid being exposed to the particulate matter in the air. We do have protective mechanisms in there really aimed a lot at microbes, and we're less good, I think, at protecting ourselves from pollution, which I think is what you're alluding to. And actually some of the work that's been done in the UK is marrying up nicely with some of the work that I'm doing, which is looking at how people who've never smoked go on to develop lung cancer. And it seems clear, again, demonstrating the overlap with immunology, that an individual has, well, there are variations in how people deal with inflammation. And I think we can instinctively see that because there are some people who are more predisposed to have things like asthma or emphysema or other types of diseases. And I think developing lung cancer in some people will have a similar mechanism, that it's down to your immune system and how it reacts to the environment.
And needless to say, lung cancer is often a particularly aggressive disease. Amelia, do you want to elaborate on why that is and what research is underway to tackle that?
Yeah, I think from what Venessa said, there is a large body of work now trying to understand more deeply the role of the immune microenvironment in looking for those rogue cancer cells and eliminating them before a cancer develops in the first place. And typically in the past, unfortunately, lung cancer would often be asymptomatic until patients had quite advanced disease. And this meant that by the time a patient was diagnosed, they had very limited treatment options. That's now changing. People are being diagnosed earlier, which also gives them more opportunities for better treatments as well. And I think the research now is trying to understand how do we support people who have very early stage cancers and the best treatments at that particular stage of their cancer so that they get the best possible outcome with minimum side effects. And I think research that's now looking at human tumours that we're able to access is giving us much deeper insight than we've been able to get from the models that we've had to resort to in the past. And I think the research in developing more clinically relevant models is really enabling the research to understand the biology of the disease in much more detail and to reveal better treatment strategies.
And Amelia, maybe you can just elaborate a little bit on your work on the matrix. What is the matrix and why is this important in cancer?
Yeah, so the matrix is not a blue pill or a red pill decision. The matrix actually refers to what we call the extracellular matrix, which is the protein that surrounds the tumour. So all organs have this protein-based macromolecular network that creates the organ structure or the shape of it. And the cells are attached to this matrix and they sense what type of matrix is there. And that can tell them to become a heart cell, become a muscle cell, or in the lung to become a particular type of lung cell. And so normal lung cells are interacting with this matrix, but so are cancer cells. And we now understand through the work that we're doing looking at human tumours that the presence of the cancer cells is actually changing the nature of the lung matrix. And we've identified some key features that are making these tumours more aggressive and enabling these cancer cells to spread to other parts of the body. So where normal chemotherapy treatment targets the rapidly dividing cancer cells, it's still leaving this matrix in place that's enabling the support for existing cancer cells that aren't quite killed by the chemotherapy. And we're trying to devise new ways to target these matrix changes in the hope that we can combine that with chemotherapy treatment to more effectively treat the tumour as a whole. So it's about looking at the environment that the tumour is in and trying to normalise that environment back to healthy tissue to have the best success of treating the cancer.
Amazing, amazing. Any other questions? Great. How did we go in answering your first question?
Sorry, yeah, I have another question. I have watched the news that, as far as I remember, Professor Valenzuela, he has isolated stem cell from hair follicle, and then he injected the stem cell. Sorry, like the he isolated the stem cell from human follicle, and from there he has generated neuronal cell, and then he injected that neuronal cell into the brain of a dog, and he observed that it works, like the neuron regeneration is possible. So using the same approach, like the cell therapy, is it possible like to isolate the stem cell from lung tissue and then to treat the COVID patients, something like that? Any research in advance or is undergoing?
It's a really interesting question. So essentially, the question is, you know, how are stem cells applicable in developing new cellular therapies, and you know, particularly potentially for the lung? Venessa, did you want to have a crack at that one?
Yeah, so I guess there's nothing that I know of where that kind of therapy is being used to generate new lung tissue, but in a similar vein, we are using it. It's still in sort of clinical trials at the moment for treating patients with solid organ malignancies, and perhaps a little bit ahead in the queue is hematological malignancies, but using T cells. So T cells are an immune cell, and there are lots of ways that this is being used, but one of the ways that they're doing it is they're extracting T cells from patients. They're exposing those T cells specifically to cancer cells in the hope that those T cells will learn what those cancer cells look like and be more effective at killing them. They're then proliferating those T cells in the lab, so getting a whole big bunch of them and then infusing them back into the patient. And so I think that's a little bit sort of what you were trying to say about, you know, harnessing our own cells to treat self. As far as I know, that's sort of the furthest along in the clinic, and I'm sure Elissa will have some insights on that. But exactly what you were saying, I don't think that's really hit trials yet. I know that it's being done in the scientific field.
And certainly, I mean, the great thing about the immune system is that it's easy to get immune stem cells. So that's what when you've heard about bone marrow transplants, that's what all the stem cells that lead to generation of your immune system are in the bone marrow, or actually now they have techniques so that they can just actually extract them from people's blood. So you no longer need to donate bone marrow, you can just kind of donate your blood. And the great thing about that is that actually now people are trying technologies for people who have genetic conditions that affect their immune system. You can take out their immune stem cells, potentially do genetic engineering on them so that you can fix that genetic mistake that they have in their immune cells and then put them back into that patient. You ablate their immune system, so get rid of their immune cells, and then these stem cells form a new immune system that's now hopefully fixed and can cure that person's disease, which is what we've been doing with bone marrow transplants in patients for a long time. But then you had to try and find a matched donor who you can then donate it. Now people are working on genetic technology so you can take your cells, fix them, put them back in and hopefully cure you.
It's just amazing. I don't think we can even begin to imagine what the future looks like in the way that we treat disease. Yes, Rory.
Just a quick question. I gather that the Garvan Institute produces phenomenal information and analysis. Is there an active interface, if that's the right word, between the Garvan Institute and industry where drugs are produced, so that all the information that you produce is provided and targeted to the efficient? And what would be the nature of that interface, if it does exist?
Great question. Sarah, do you want to comment on how that works in practice?
Sure. So we can't do it wholesale. We can't sort of say, here's all our data and share it, because we have responsibilities for the patients that different studies, patients will have made different decisions about how they want their data shared or not shared. However, we do actually have a lot of close connections with industry and drug development. So we have examples where our researchers have developed molecules or strategies for treatment that are now in clinical trials. So we do some of that in collaboration, but driven by Garvan. We also have lots of examples where we collaborate with industry to provide a piece of the puzzle that's needed for them to then go run trials. But it's more study by study than wholesale. That said, a project we're working on now on the data science side is to improve the way that we store and are able to share our data, so that at least if research participants have said, yes, we're happy for data to be shared, that then it's a very easy click of a button to make, on the technical side, to make that happen. Because historically, studies were sort of siloed, and we're trying to bring data together to make it easier to share. But I think there's still going to be a need to think about it study by study, just to make sure that we follow what our research participants have requested.
Fantastic. Now, everyone, we've got a special surprise guest. I can see loitering at the back of the auditorium. Professor Chris Goodenow, come on down. We've got a really special announcement to make today. Many of you will know Chris as our, unfortunately, very recently outgoing executive director, but fortunately for us, Chris has returned to the laboratory and is continuing to undertake really important work in particularly autoimmunity. Now, on today, International Women's Day, Chris has given us approval to make a special announcement about a contribution that he is making with his family to the Garvan Institute, very specifically for our women scientists. So ladies, this is a surprise even for you, but you'll hear it here first. So Chris, come on down. I am going to put you on the spot, but first of all, I'm just going to say a few words about this special initiative. We've heard the incredible talent that we have here from today's presenters, and there is incredible talent right across Garvan and right across this precinct and this campus. And so we're always looking at ways that we can increase support, particularly of our women scientists, and increase diversity and inclusion within the Garvan Institute and with our partners. So Chris and his wonderful wife, Dr. Suzanne Hartley, have very recently made an incredible donation of $1.4 million to establish the Jacqueline Goodenow and Barbara Hartley Prize here at the Garvan Institute. Chris, I'm going to get you to just tell us a little bit about the mums. You can come up. Can I get a round of applause? So, Chris, what inspired you to establish this prize? And tell us about Barbara and tell us about Jacqueline, because they were phenomenal women and produced this incredible human being and science brain.
Oh, well, you know, we're completely off script here. I'm not even meant to be here. I was just peeking down the back to hear what these brilliant scientists here had to say. And so I don't want to steal your show. Suzanne and I, had this opportunity to make a gift, and we thought, well, we want it to be for Garvan, but we recognised that there's one of the big challenges that faces all scientists is this tension between wanting to take the risky path that's less well-trodden or sticking to the well-trodden path. Now, unfortunately, particularly these days more than ever, a lot of the financial rewards that renew your salary and your grants and allow you to support your lab and your family and everything else, they're pinned to the number of publications in Prada-branded journals, if you like, that you've had in the last couple of years. And all that tends to discourage scientists from tackling the really hard problems that everyone else says, oh, that's too hard, because that will mean less publications and it might, put in jeopardy whether you can pay the mortgage. That applies for every scientist, but I think you all know for a variety of reasons, those obstacles are greater for women. And so that was the thing that we thought, well, what could we do to at least lessen those obstacles a little bit? And so we were very conscious of the fact that both my mother, Jacqueline, and Suzanne's mother, Barbara, had faced those obstacles in different ways. Perhaps the most extreme way was my mum graduated from Sydney University with a university medal in psychology, had suddenly discovered, oh, I love this science business, having you know, had a scholarship at down the road at St. Vincent's College for Women, but there was no science on the curriculum because that wasn't something that was going to be a path for women in those days. And so she discovered she loved science, and of course it was right after World War II, and they desperately needed people to run the course, and she was running the course. And so she knew there was a job coming up as a lecturer. Thought, well, I'm doing the job, and everyone says I'm doing it well, I'll get the job. And someone had to take her aside and say, it's only going to be a man that gets the job. We don't have female academics in Australia, and if you want to pursue a scientific career, you're going to have to go to the US. So she was lucky to get a philanthropic grant, a scholarship to go to Harvard, and did a PhD there. And anyhow.
And met Chris's dad and Chris's sister.
We came into an opportunity to be able to give back a little, and we're just thrilled that we can give back in a way that, you know, I have seen in my career with some of my female trainees. They get a prize, and it just frees them up a little bit to do whatever it is that they need, that allows them to take a few more risks and just take that pressure off so that they can be more creative as a scientist and stay in science. So
amazing. Chris, thank you so much, and thank you for letting me drag you up here. Really, really special. To our scientific superstars, thank you so much for sharing your work, of your lives, your brains, and your passion for what you do. We are so privileged to have you at Garvan, and we're so privileged to have had you take the time to speak to both our audience in the auditorium and those in the Zoom room. So really, please, thank you so much. To our lovely audience, thank you for joining us. If you have enjoyed today, please do recommend us to your friends and family. Tell them to jump online and watch this seminar once it's available, or any other seminars. Encourage them to get involved in being part of the Garvan family. So much of what we do is enriched by each of you, your support, your passion, your philanthropy. I know that we have a number of our special partners for the future in the room. Hello, our wonderful partners. Thank you for being here. So hope to see you at the next one. Now, we will be introducing Chris's enduring replacement, Professor Benjamin Kyle. We haven't set the date yet, but stay tuned, and I really hope you can come and meet Ben and hear what he has in store for the next period, the next 10 years of Garvan's exciting future. I think it's only going to get more and more innovative and have more and more clinical impacts, particularly when you hear about the incredible work that these guys are doing. They are going to change health outcomes for us all. Thank you. Great to see you.